Have you ever wondered about the 40-year rejection? How Catalin Carrico believed synthetic mRNA could cure almost any disease, but universities demoted her because nobody wanted to fund it. The problem was simple. Synthetic mRNA kept killing lab mice. Their immune systems saw the foreign genetic code as invaders. She was practically evicted from her lab, surviving on photocopiers. Then, a chance encounter at a copy machine changed history. She met immunologist Drew Weissman, who needed a vaccine breakthrough. They discovered that swapping one chemical letter tricked the body. That single, overlooked adjustment laid the foundation for global rescue. In 1985, Carrico sold her car, hiding cash inside a teddy. She landed at Temple University, ready to test synthetic mRNA. Her goal was teaching cells to make their own medicine, but injected mRNA triggered severe, lethal inflammation in laboratory mice. Skeptical review boards repeatedly rejected her grant applications for funding. University bosses saw her research as a dead-end money pit. In 1995, Penn demoted her, slashing her salary drastically. She lacked a permanent position and had no lab assistants. Working there felt like building spacecraft out of wet cardboard. Then, a chance meeting at a photocopier changed medical history. Penn repeatedly rejected her grant proposals for mRNA research. Then, a chance meeting at a copy machine changed everything. Immunologist Drew Weissman wanted a better way to make vaccines. They joined forces to make mRNA work inside living cells. But their early tests triggered massive, deadly immune reactions. The lab mice puffed up like tiny, angry cotton balls. The body's defense system recognized the synthetic RNA as hostile. They compared it to natural RNA to spot the difference. Natural RNA contained modified nucleosides that bypassed the alarm. By swapping one building block, they hid mRNA from sensors. By 1995, UPenn issued an ultimatum: demotion or leave. She chose demotion, losing her lab space and salary. The science itself had hit a massive, seemingly fatal wall. Synthetic mRNA triggered severe, lethal inflammation in test mice. It was like trying to deliver medicine wrapped in fire. Other researchers abandoned the molecule, calling it too dangerous. Then, a chance encounter at a shared office photocopier changed everything. Immunologist Drew Weissman needed a way to make HIV vaccines. Together, they tackled the deadly immune reaction that stopped everyone else. She kept working anyway, refusing to let her research die. Then, a chance meeting at a photocopier changed everything. Immunologist Drew Weissman wanted to make a new vaccine, but their early mRNA tests triggered deadly immune reactions. The body saw the synthetic RNA as a hostile invader. They spent years testing every single piece of the molecule. Finally, they swapped out one single chemical letter called uridine. This tiny tweak let the mRNA slip past immune defenses. It worked perfectly, producing proteins without causing any harm. Then, a chance meeting at a photocopy machine changed everything. Drew Weissman joined her, but their injected RNA triggered fatal inflammation. They swapped one chemical letter, creating a stealthy hybrid molecule. Suddenly, the body's defense system let the mRNA pass safely. They published this crucial discovery, expecting the world to notice. Hit the subscribe button and press the bell icon to get updates. Instead, the scientific community greeted their paper with total silence. No grants arrived, and Carrico remained on the academic sidelines. Yet, two obscure startups named Mrna and BioNTech quietly licensed it. Then, in late 2019, a novel coronavirus began spreading globally. Traditional vaccine methods were simply too slow to stop it. Researchers quickly turned to Carrico's overlooked, modified mRNA technology. Scientists designed the vaccine's genetic template in just days. They packaged the delicate genetic instructions inside tiny fat bubbles. Clinical trials soon revealed an astonishing 95% efficacy rate. Millions of doses shipped out to clinics around the world. The scientist who faced constant rejection suddenly became a hero. Her 40 years of stubborn persistence finally saved the world. In early 2020, a novel coronavirus shut down the entire world. Scientists instantly turned to Carrico's modified mRNA technology for help. They designed the highly effective vaccine sequence in just hours. Clinical trials proved the new vaccine was 95% effective. It shattered old vaccine speed records like a sports car. Billions of doses shipped globally, saving millions of human lives. In 2023, Carrico received the Nobel Prize in Medicine. The university that once demoted her now celebrated her triumph. Today, her mRNA code targets cancer, malaria, and future diseases.